So phylogenetic analysis, protocol and interpretation of results. Now since last 6 days we are working with various, what are the methods of phylogenetic analysis, which data types that can be used for phylogenetic analysis, and which are the techniques or which are the algorithms that are available and which are suitable, most suitable. So that we are going through. You worked a lot, exhaustive practicals were given to you for analyzing a data. So we tried, means accordingly the tutorials were designed so that it will be easy for you. You can understand the software, you can go through it, so that was designed accordingly. Then also it was necessary part, okay, one should have huge or high amount of expertise, practical session. Hands-on training should be more, or for maximum hours of time. So that was the main purpose, and I tried and given that time to you rather than skipping the time for a lectures. Now it's a part you worked with sequence data, that is protein, nucleotide. You worked with RAPD or molecular type data, means RFLP, RAPD and ARDRA. In that case, we have given one paper also, that is Professor B.N. Jory's paper based on diversity of rhizobium, whatever the isolates have collected, and that was the scientific approach we have given, so that it will be useful to you how you can use these techniques of phylogenetic analysis in analysis of data. And then also I designed two morphological data analysis that I have given, one based on what the weather means where both the alleles are present on the same locus, and another one that is based on the alleles which are present on either of the alleles, one of the alleles from that, both the alleles there are allelomorphs is absent in that locus. Now these were computational methods we have studied. Methods in phylogenetic, that is tree construction method. One is evolutionary distances or distance method that we have seen. The distances are calculated, that is from pairwise comparison of sequences, forming a distance matrix which is used in subsequent analysis. Main purpose, we will try to compare what was a pairwise alignment of the sequences. Sequence will be pairwise alignment. What is pairwise alignment? Okay, we will compare a character of one sequence with the character of other sequence. Then while comparing the character of one sequence with the other, there will be the match, mismatch or completely mismatch. Or match in case of protein or amino acids, there are 20 amino acids are there. So probability means, okay, matching, we have to consider the physico-chemical matching also. A match, alanine matches with alanine, that's a complete match. But when alanine matches with valine, that is mismatch, but the scoring may be positive there because of the physico-chemical properties. So that's the thing. So that score is considered for a distance matrix creation. In case of RAPD data or gene frequency data, the data set is very small, but in case of the sequences, the data set is more, or large number of characters are there, means 1500 on that much. Okay. So that's what key sequence data type is more feasible, or that is useful for carrying out the phylogenetic analysis or grouping of organisms. So number of characters you have, so that proves or that gives a more valid base for a construction of phylogenetic analysis or grouping of organisms. Now cluster algorithms, so that was given by actually Everett 2001, that is mostly used now, distance matrix into a phylogenetic tree. Now most popular distance methods are UPGA and neighborhood joining method. You have tried both these method. Okay. One another modification of UPGA is nearest neighbor, and that you can find in past software. Paired and neighbor or single linkage. Neighborhood joining is another clustering. Okay. So these are the two methods. Then in case of character strength method, parsimony, maximum parsimony, that is minimum number of changes. Okay, it will draw out all the possible tree, out of that which is having a minimum evaluation or minimum changes will be selected, maximum likelihood, log likelihood score optimized over the branch length, means multiple sequence alignment will be done. Then again that same thing, informative and non-informative sites will be found out. Then number of possible tree will be drawn. But next thing, a likelihood score will be calculated. Likelihood score that will be based on what is the either transversion or that is replacement. Okay, either transversion or replacements, maybe that things. Okay, score will be given. So already a score is already there in the algorithm. That means what is the probability that one character will be changed by other character. That's why this is very, means the parsimony, though it's also exhaustive, but maximum likelihood method is more exhaustive as compared to the parsimony. Parsimony method is just try to find out the parsimonious tree, whereas maximum likelihood method, it goes for the parsimonious as well as it depends on the number of means that is based on the likelihood score or likelihood score of a tree. Okay, so with having a maximum or minimum evaluation over there and maximum likelihood score, so that will be selected in case of the maximum likelihood method. Then minimum evaluation tree length is the okay Bayesian method, these are not, but this is the highly preferred. Now if we talk about the Bayesian statistics probabilistic method, this is the best one, it's a recent. This method is best, but whatever knowledge or references or literature is saying that, so okay, these are highly used method, a systematist. They will always go for a maximum parsimony method and or maximum likelihood method. But the drawback of these two methods is that if you try to use more than 12 or as you increase the number of tags in the analysis, what will happen? It becomes more and more exhaustive. The number of trees, the probable trees will be more. That will increase, and what will happen? The analysis, that will be the slow process, means it will be a very slow process and exhaustive. Okay, so this, in this case maximum, if you are working with the 10 or 12, then it will work faster. You can go for a 20 what you can say 20 taxons also, or even 25. But if you are working with the more than 25, try to avoid. It may show or it will be get slower. Or if you are working for, means then if you are replicating the data, replication means again your bootstrapping or jackknife you are using, then that time it will become more slower. You know that how you worked with the bootstrap or replication of data, you have to wait for, because if you are going for 1000 replications, so it was a time consuming. So this method again time consuming because it will draw on number of possible trees. Now in case of these also, these are faster, that's what. Now this was the MS in file format. What are the errors? Means more, I have seen that everybody have drawn the calculated the MSA, but what the mistakes they did? Okay, when you get or retrieve the sequences in FASTA format, you just know okay there is a greater than arrow, and after that the GI number starts. But it's also, we always want okay there should be an organism name. So try to convert this Clostridium 1, okay, or write CL, C underscore species name, so that you can get a good. So everybody tried okay Clostridium. What happens while you retrieve the multiple sequence alignment in file format? It always takes the first 10 characters after greater than arrow. So that's error. So I changed this yesterday. Again, miss you can do this after okay after retrieving the alignment. Okay, so you want to change the names, but you should keep in keep in mind that so okay there should be a 10 characters, spacing should be 10 character. Okay, so like Clostridia underscore 1 I did. So 1 2 3 4 zata. So 1 2 3 1 2 3 Bacillus subtilis. One was actually Bacillus licheniformis and amylolicheniformis. That was the species. Then don't disturb this alignment. You can also check okay this the last character should be in one column. So if whatever if you place one character ahead in between, or if it goes beyond the 10 character, then also you will come to know it's okay that character has been pushed forward or by one character. but the naming should be proper, so that for construction of the dendrogram it will be very easy. You can also do the things, or you can also change in you can also edit the tree file, but it's very difficult when you get 3 Clostridia species in the one tree. So better you do it before means generate the file, input file with a different organism name or specific.